It's my pleasure to introduce our speaker for the Physiology Seminar Series today, Glenn Lyszczak from the Biochemistry Department here at UT Southwestern. Glenn started off during his PhD at UPenn, studying the structure and functional mechanisms that underlie the activity of amino-terminal acyltransferases. From then, he went on to a postdoc with Tom Muir at Princeton, and he took on a couple of really big questions in the chromatin field, which are, the effects of PARP enzymes and the effects of ADP ribosylation. He started looking at the specificity of ADP ribosylation in relationship to histone acetylation. And he's made a number of important insights in that area, looking at the DNA damage response. More recently, he's been developing tools to address another big question in the PARP field, which is looking at the differential effects of AB ribosylation chains of different lengths. And he's developed some really nice biochemical approaches to kind of address these questions. With that, I'll turn things over to Glenn.
All right, thanks, Matt. Let's see. You see the slides?
Yeah, we do.
And laser pointer. All right. So yeah, thanks for the introduction and for the invite here today. I'll be telling you a little about our work with ADP ribosylation in my lab in a talk that I call New Technologies to Study Chemically Complex Protein Modifications in Cellular Signaling. And so I like to start by just defining what I think of as a chemically complex protein modification. So here on the left part of your screen, we have what I would consider chemically simple modifications. These are modifications that I would hope that first-year grad students would be able to draw on a board in the first class. Just small chemical adducts, a few atoms that modify the side chains of, for example, lysines or serines, things like phosphorylation or acetylation, which can change the charge of these side chains and have quite impactful effects on cellular signaling, as many of us know. We think of things like small proteins, ubiquitin-like proteins, which clearly there's not much need to explain the biology of these modifications, but these are chemically complex, right? They're folded proteins, large polypeptides attached to side chains of other proteins through what are called isopeptide bonds. And we have really good tools to study these complex modifications. And as a result, we've learned a lot about biology and disease, and even more recently, a movement into exploiting these pathways for therapeutics. And then for chemically complex, I'm showing you here this mark ADP ribosylation, which has a long history, I guess, internationally, as well as here at UT Southwestern. And this is where the ADP ribose moiety from the NAD cofactor is transferred to the side chains, to many different chemical side chains on proteins. Here I'm showing you a serine link, can also be on aspartate and glutamate residues, arginine residues, glutamine residues, so all sorts of different residues. You can see what the structure is here. This is for a mono ADP ribose unit, but this mark can also be polymerized, much like ubiquitin, where the mark itself serves as a substrate for an additional mark. Here I'm showing you what's called linear polymerization, which would occur off of this hydroxylate circled here. There can also be branched polymerization, where it occurs off of a difference. So this can be very topologically complex. And again, we come back to this chemically complex. This mark is quite difficult to synthesize chemically. So as opposed to something like acetylation or phosphorylation, where you can just buy modified amino acid building blocks, build those into peptides to study their function, you can't do this with ADP ribosylation. And it can also be quite unstable. So certain marks are very easy to hydrolyze. They're quickly turned over in the cell. Often they're not amenable to traditional mass spec proteomics methods. And as a result, there's currently really only two site-specific antibodies that were developed incredibly recently. There's other pan detection reagents that are quite effective to look at sort of the ADP ribosylome in general and something like a Western blot or immunofluorescence. And there's been lots and lots of sites identified or reported. And there are debates about how many of these are real and what are the biological consequences of these. And so it would be quite helpful if we could find additional tools to study such marks. And I think, we think of ADP ribosylation. There's also a lot of other marks that I would consider to be chemically pretty complicated, right? So where we have these small molecules that are appended to the sides of proteins, something like serotonin or just amino acids that again can get conjugated to proteins. So this is happening all over. And again, the tools in these fields are also progressing quite nicely right now. And as a result, our understanding of the biology is improving greatly. Okay, so these are very straightforward concepts that we have in our lab and very straightforward questions, right? So here I'm just showing you some enzyme. In this case, it's a PARP enzyme, or the enzymes that install ADP ribosylation. Showing you a substrate. In this case, it's the nucleosome, right? So the DNA wrapped around that core of 8 histone proteins, so 4 unique histones, 2 copies of each. Enzyme finds substrate, installs a modification, and there's a signal. And so on the left, we're interested in things such as how are these modifications initiated, right? So what stimuli start these cascades, bring the enzyme and the substrate together? How do the enzymes work? So just mechanistically, as an enzymologist, right? What is the mechanism? Are there auxiliary factors that can control the activity of the catalytic subunits? And what are the substrate modification sites and how is selectivity achieved? So what residues are getting hit and how does the enzyme know that those are the residues to hit? And so we use, much like a lot of other people on this call, very standard approaches in this area: genetics, biochemistry, small molecule probes, proteomics, et cetera. And a bit of our story will touch on the left hand, and the rest will touch on the right hand panel here that's now circled, which is how do these modifications affect target protein function? And so we know that protein modifications can have all sorts of biological effects as listed here. And we want to study these. Typically we'll inactivate the enzyme that installs the modification or mutate the target site on the substrate protein. So block the modification from happening in some way. These can have many, many effects, be them direct or indirect, right? They can have many off-target effects, et cetera. So it's very hard to establish a cause and effect relationship, right? PTM X causes function Y. And a lot of other major challenges in this area come from the fact that these PTM cascades tend to be very rapid, particularly in the DNA damage response, which is where this PARP1 signaling that I will talk about today is occurring. So they're very, very fast. They're on and off, quite low abundance. So it's not that every single protein in a cell gets modified. Often it's a very small percent of the total population in the cell that's getting modified. And they can be quite combinatorial. In particular, in the case of nucleosomes, this means there can be many, many modifications on a single protein. Again, making it difficult to discern which modifications are essential for a given signaling event. Okay, so that kind of launches me into the system that I'll talk about today. So here I'm just showing you a very simple cartoon of DNA damage-induced ADP ribosylation. So here I'm showing you PARP1. PARP1 also has a paralog PARP2 that I won't talk about much today, but a very similar function. These enzymes have a DNA binding domain or a nucleic acid binding domain that can recognize DNA strand breaks. They bind to DNA strand breaks that triggers NAD binding. So they can't bind NAD until the nucleic acid domain is properly bound. Binding domain is properly bound. Once that occurs, they can bind NAD and they begin to ribosylate proteins in the area. And I'll show you a Western on the next slide, but they can hit lots and lots of proteins. But typically on that Western, you'll see substrates jump out, PARP1, so modifies itself, auto modification, and nucleosomes. Again, this is what that modification looks like. And so if you damage cells, so you just expose, in this case, 293T cells to a damaging agent, this is what you'll see. So you do see this background ADP ribosylation in the undamaged. But again, you see lots of these substrates popping up, primarily PARP1 and those histones. And so there's a lot of discussion about what are the relevant chain lengths These can be up to 200 units in length. They could be relatively short, 1 to 10 units in length. And so if you kind of look at this total pool from cells of just poly ADP ribose chain length, this 1 to 10 area is highly abundant, and a lot of readers, most of these sort of bona fide ADP ribose readers, are going to recognize somewhere from that 1 to 10 region in length. So as Matt said, we've developed technologies. We think that that 1 to 10 area should let us cover a good bit of these signaling events. And there are currently 4 FDA-approved PARP inhibitors. These are for DNA repair deficient cancers. They're being tested in several other diseases right now, things like neurodegeneration, cardiovascular disease. And as of last week, I think I looked this up, and there's 68 active clinical trials for PARP inhibitors right now. So again, there's a lot of activity in this field at the moment. Okay, so recent progress in the field has revolved a lot around PARP1 activation and substrate identification, figuring out what all these bands are. And our goal is to fill in a gap, which is deciphering the downstream signaling consequences of specific ADP ribosylation events. I would say comparatively, there's a lot less progress in this area. And so the reason we're interested in that, and I'll get into this towards the end of the talk, is because beyond just learning about physiological function of this mark, we feel that this can be very useful to help to inform PARP inhibitor susceptibility, so potentially identify new biomarkers for PARP inhibitor sensitive cancers, or how to overcome PARP inhibitor resistance mechanisms. And our starting point, so the first proteins that we were interested in were nucleosomes, so histones. And again, that's because we just kind of picked out what are the most abundant ones on the gel. And I was trained in epigenetics and nucleosomes, and so it's really a great match for my skill set.
Hey, Glenn.
Yes.
I had one question. So with relationship to the fact that these enzymes require NAD, so can NAD redox homeostasis change, because it's very dynamic during, like, in response to stress, under different metabolic conditions. I'm just wondering, can the NAD limit chain length, or does it just affect total activity of the enzyme?
Yeah, so the enzyme, and a lot of people have done a lot of things with playing with NAD. Lee Krause here at Southwestern is really into this area, knocking out the NMNAT proteins and showing that they're incredibly important for PARP function. For us, it seems like PARP will burn NAD if you sort of expose cells to a lot of UV light or put peroxide on cells. PARP will basically run through that NAD until it's almost not detectable by sort of commercially available NAD detection kits. So I think that NAD availability is very important here. Yeah. I don't know how physiologically relevant that is most of the time. I think a lot of damage events, they're not the bomb that we're putting on them for things like that.
Yep. Thanks.
So the place that we started here was, and this was back when I was a postdoc, I'll just kind of give you this because it's a lead into what I've done here at Southwestern, was just trying to reconstitute nucleosome ADP ribosylation. It's a very simple assay where we can purify all the individual components, so all the histones and nucleic acid, to make recombinant mononucleosomes. And then we can mix that with NAD and recombinant PARP that's purified from insect cells. And just the readout is, do the nucleosomes get ribosylated, right? And so doing this, we saw what I think a lot of other labs had seen for a very long time, several decades, which is you see a lot of PARP autoribosylation. So the PARP is active, it's modifying itself, but it doesn't look like it's really touching the histones all that much. And so I think this was kind of a bottleneck in the DNA damage induced ADP ribosylation field. So a lot of people were kind of trying to figure out what the sites are, and you'll see very low abundant aspartate and glutamate. Other papers were reporting lysine ribosylation in this assay. So there was a lot of confusion, I think. And it was around this time that a paper came out identifying a PARP1 activating factor called HPF1, or histone parylation factor 1. And there are a lot of PARP enzymes, PARP binding proteins that had been reported in the past. So I think you never know how much to read into these, but I think this paper is very compelling. I made an HPF1 knockout cell line. You can see here that when you knock out HPF1, you no longer see DNA damage-induced ADP ribosylation, very consistent with what this paper was saying. A follow-up paper came out shortly after saying the modification was happening on serine residues in the context of DNA damage. And so on the strength of these stories, I decided to purify that HPF1 component in E. coli and just ask if I throw it into my nucleosome ribosylation assays, what happens? And the blot lights up. So you get lots and lots of activity on your histones now, even on that activating protein. And so I went through and scanned all the serine sites in the nucleosome and found that it's hitting this S10 residue, very little bit on this S28 residue in the H3 tail. There was another site on another tail that I'm not showing. But if you go through these sites, and again, you serially mutate them, what we found was that S10 residue was really one of the primary sites of nucleosome ADP ribosylation. We could then repeat these assays in the mammalian cell, where we introduce flag-tagged H3 transgenes into the cells. We let the cells incorporate those into nucleosomes, damage those cells, and then IP those histones back out. And what we find is that, yes, we can detect damage-dependent ribosylation, and if we get rid of that S10 residue, we no longer see that ribosylation. So indeed, that seems to be the target site. So really, there's two target sites, that H3S10, and then another histone. Again, it's sort of a similar move that we had done. We also found this H2BS6. So these two sites being the primary sites for HPF-stimulated chromatin ribosylation. And this has also been shown by some of these other labs that I had reported on the previous slide. So right around this time, there were about 2 or 3 papers that came out, all reporting these sites. So we're quite confident these are the correct sites. So then the next question became, and this is kind of when I got to Southwestern, we now know what the target sites are in the DNA damage response, but how are they stimulating DNA repair? And so we were starting from a point where we know, and we've known for a very long time, many decades, that PARP1 activity correlates with DNA accessibility at damage sites and repair protein recruitment. Okay, so the challenges are, again, as I said sort of in the introduction, it's not really possible to mutate these histone target sites in mammalian cells. So that type of genetics, cells aren't viable, but it's really hard to do. There's lots and lots of histone genes in your cells. And again, knockout of PARP and HPF affects many, many substrates, likely dozens to over 100 ADP ribosylation events are going to be misregulated. So again, pinning it down to our specific sites and what they're doing would be very challenging in that way. So again, as I said before, our plan was to prepare semi-synthetic ADP ribosylated nucleosomes. So can we make nucleosomes, right, on the benchtop and install these modification sites, right, specifically where we want them, and also control ADP ribose chain length. So the way that we do that is through synthetic protein chemistry. So synthetic protein chemistry involves synthesizing fragments of proteins, so synthesizing peptides completely in the flask. So we're not dependent upon the ribosome or the genetic code. We can put in whatever we want. We can put in modified amino acids. We could put in canonical amino acids. We could put in mixtures of different modified amino acids. So we have complete control over chemical composition, over where those modifications are going, and everything comes out very homogeneous. So we're 100% what we think we are. And then if we synthesize this peptide with a handle called a C-terminal thioester, okay, this C-terminal thioester reacts with N-terminal cysteines to form a native peptide bond. Okay, so we can express the remaining bits of a protein in E. coli, something very large. So this lets us get around the size limitations that are imposed in peptide synthesis to create a full-length protein. And so you can also do this iteratively, so we can fuse things to the front end, we can fuse things to the back end, we can do multiple ligations. So really we can kind of piece together proteins to form full-length proteins, again, that are scarless. So now we just have a modified protein with no other strange chemistry going on in there. And these methods have been around for a long time, and they've been instrumental for studying things like acetylation, phosphorylation, ubiquitination, all these marks we've talked about before. But again, the challenge is, how do you get ADP ribose onto the peptide? So it turns out ADP ribose is not stable to most of these classic peptide synthesis procedures. And we had tried this for a while, about a year, with the chemist here at Southwestern, and we can get the ADP ribose onto more simple peptides. And that has been done in the past. But to get it onto a peptide like histone H3, which is a bit more complex in terms of the side chains and the chemistry that goes into the peptides, it was never a go. So I'm going to tell you about our technology here that was performed primarily by a graduate student, Jugal Mohapatra, who you see on the bottom left of your screen. And what Jugal figured out we could do is that we can make these synthetic peptides. Okay, and we could mix them in with a reconstituted PARP-HPF reaction. And then if we ran an HPLC, we would see unmodified peptide, we'd see monoribosylated peptide, and then we would see different chain lengths coming off of this peptide, and then he could separate those. And so that's kind of the overview, and now I'm going to go through it just a little bit more in detail. So we take a substrate peptide. Again, this is that H3 tail sequence, right? And there's that KS site that we found gets modified in the context of the nucleosome. And we mix it with just PARP, so no HPF. You can see we really get no modification, just the unmodified peptide. Now, when we put this HPF protein into this reaction, and we run this reverse phase HPLC column to analyze the products, we see a peak that corresponds to our peptide plus 1 ADP ribose, our peptide plus 2, plus 3, plus 4, plus 5, and so on. And we can collect all these peaks separately and analyze them by mass, which is what I'm showing here. And if you're not used to looking at peptide mass specs, these are quite clean. So the separation was very efficient. So what we knew was we're putting ADP ribose on. These are all some derivative of a plus 541. And we're pretty sure we're putting it at the KS, but how do we really validate that? So the first thing we did, and this is just the same HPLC trace I was showing on the previous slide, is we took our reaction, we used the S10 to A, so we mutated that target site. We got rid of all activity. So that's definitely important for the activity. And then the last thing we wanted to verify was, is this definitely all coming from that one site? Or is it that S10 gets modified and that stimulates modification of other residues? And so to do that, we purified these glycohydrolases. So these are very well characterized enzymes that remove ADP ribose. So PARG, which is poly-ADP ribose glycohydrolase, is an enzyme that dials poly-ADP ribose chains down to mono-ADP ribose. So it cannot remove that last group, but it removes everything else. ARH3 is an enzyme that can hydrolyze ADP ribose from serine residues. So our thought was, if we take these long chains and we treat them with PARG, we should come back to mono-ADP ribose, if indeed the chain's emanating from a single site, which we do. And then ARH3, being the serine ADP ribosyl hydrolase, is able to bring us down to no modification. And so all this is just to very carefully validate that, yes, we're doing this with an enzyme, but we're definitely putting it exactly where we think we're putting it. All of the stereochemistry of these ADP ribose units is correct. Site specificity is there. And we perform a different type of assay that I'm not showing you here. It's an LCMS MS-based assay to validate that these chains are principally linear. So I pointed out that modification can occur from those two different hydroxyl groups, either linear or branched. But ours is, I think it's like 99.98% linear by our LCMS MS assay. So again, we're putting linear chains, we know where we're putting them. And for every peptide that we make, we do this type of characterization to make sure that it's correct. So the next problem that we had was that we can perform this, we can separate our peptides. We were not able to scale up to get milligram quantities of these different poly-ADP ribosylated products. And so the solution, we thought at the time, was, well, we could just bomb in more HPF, more of the activator would get more activity. And so we thought this because all these previous papers about HPF had said, if you have HPF in cells, you get large quantities of shorter ADP ribose chains. And so we tried this, And what we ended up seeing was as we increased HPF, so this is the unmodified peptide, this is the mono ditri, we're seeing more and more of the mono, less and less of the poly. And so what it turned out we were looking at was HPF is converting PARP into a mono ADP ribosyltransferase. So if we kind of dial back, and I'm not showing you the kinetics here, it turned out that indeed that mono ADP ribosylation reaction goes faster and faster with more HPF, whereas the elongation is basically being inhibited. And I think in retrospect, this is exactly what was supposed to happen, because we know that PARP can elongate from itself in auto ADP ribosylation reactions. We know that, we see that on gels, right? And we see in cells that HPF causes shorter chains. But what's actually happening is that this is probably a two-step mechanism, right? Meaning that this serine mono reaction, right? This priming event is performed by the complex, and the elongation reaction requires HPF1 to take off, to leave, dissociate. And then you have PARP to perform that elongation reaction. And so once we saw this, we figured what we could do is make a lot of the monopeptide, right? So use a lot of HPF in the first reaction with unmodified peptides, convert it all to mono, isolate the mono ADP ribosylated peptide, and then incubate that with PARP and no HPF, right? So just take HPF out of the equation. And would we be able to get efficient elongation? The answer is yes, that elongation reaction is very, very efficient. And so that's what I'm showing you here, that if you incubate PARP1 or that paralog PARP2 with unmodified peptide substrates, you see very, very little to basically background levels of activity at these quite modest concentrations. But if we incubate these proteins with that mono ADP ribosylated, so again, this is isolated from that first reaction, we get very efficient, greater than 80% turnover of this mono into the different poly species. And then just to kind of further interrogate this mechanism, if you take one of these reactions, for example, 5 micromolar PARP, the elongation reaction, and you incubate it with HPF, increasing concentrations of HPF, you see the HPF slowly shut down that reaction. So HPF inhibits that reaction. If you incubate this reaction with an HPF construct that cannot bind PARP, it has no effect. So indeed, HPF blocks that PARP ability to elongate. So what we found here was that serine ribosylation requires 2 unique enzymatic activities. So this is kind of that model redrawn here. And sort of around this time, the structure of HPF bound to PARP2 came out. And what the authors here found was that it looks like HPF would block the elongation mechanism by PARP2. So we know how these enzymes elongate. We kind of understand that mechanism. And what this paper had found was that the HPF would actually interfere with that mechanism. So we were happy to see that our data is very consistent with this structure. Something that we're interested in now, and that I think a lot of people are interested in, is how is this HPF1 interaction regulated, right? Because this is really going to control PAR chain lengths in cells, or at least be one of the major factors that's going to control that. So I don't have any more data on that now, but it's an interesting question that popped out of this. But for us, we were pretty excited to have our method worked out at this point. So we can now, in a scalable way, in a way that we could get multiple milligram quantities of each poly ADP ribosylated peptide, we have this method where its first step is to just use a lot of HPF1 in the reaction, get a lot of monoribosylated peptide. We could then take that peptide as a substrate, mix it with PARP, get a lot of poly, and we could separate this into different chain lengths. And so at the time, we had figured out how to do this for about 4 or 5 ADP ribose units in length. I think the longest we're at right now is 8, and we've kind of set the bar for, again, it would be great to get to about 10. So we're closing in, but everything I'll tell you about today is around 4 or 5, but that will be specified moving forward. And so we find, again, that this works very well for our target sites at H3S10. You can see these are just HPLCs with mass spec characterizations showing you these are very pure products. That S6 site works just as well. And then we took a few other known PARP HPF target sites just to show that this technology is quite broadly applicable for generating these different mono and poly ADP ribosylated peptide species. So I won't talk about any of these other proteins that we're interested in today. I'm just going to focus on the nucleosome, but that's something that some members of my lab are working on is getting outside of the nucleosome and moving into other part of HPF1 target proteins. Okay, so now we come back to, we pointed out this was the plan. So we now have a way to make these proteins. So if we mix that technology I just showed you with that protein chemistry technology, it looks a little something like this. We could make peptides that have that thioester handle, the different H3 or H2B peptides. We could get mono or the different poly ADP ribosylated peptides. We could use that native chemical ligation with the fragment of the corresponding histone. So H3 1 to 20 matched with H3 21 to 135. React them together. We have a full length ADP ribosylated H3, and then we could put that into nucleosomes. So in this way, we're able to generate a small library of ADP ribosylated nucleosomes. We could also mix and match. So I'm showing you a few of those here. We make the histones, we assemble them into nucleosomes, and then we run Western blots and native TDE gels to validate that the mark is stable and survives these processes. Indeed, it does just fine. But again, we have different H3 single modification sites. We have mixtures of H3 and H2B modification sites, different H2B modification sites, etc. Okay, so now that we have our nucleosomes, what do we do with them, right? And so we were looking for some kind of proof of concept just to get this method out. And we thought, okay, what are just known PARP-dependent processes? And I think the two that really pop up, PARP activity is known to be pretty important for chromatin decompaction at damaged sites. And so for those of you not familiar with this area, basically chromatin, particularly in inactive regions of the genome, the nucleosomes tend to be all packaged together, as you see right here. And when damage occurs in these areas, that chromatin really needs to open up. So it needs to decompact. And we know that if we use PARP inhibitors, that doesn't happen so well when you expose cells to these DNA damaging agents. And then in sort of a similar mechanism, nucleosomes are cleared from strand break sites. So this is an ATP-dependent process. These chromatin remodeler enzymes use ATP to translocate the nucleosome down DNA. What this does, if a strand break occurs near a nucleosome, those proteins will be shifted down the DNA, and you'll have, again, freedom for these DNA repair proteins to come and bind. So this is what we focused on first. There were lots of these chromatin remodeler enzymes that had been reported to be PARP dependent. And whether or not they are directly stimulated by PARP, I think for some of them it seemed like they must be, for some of them it was kind of unclear. So we purified, again, a sort of a small panel of these. I'm showing you the 4 here on the left. And we wanted to throw those into or reconstitute remodeling assays with our different nucleosomes and ask, how does nucleosome ribosylation impact the activity of these remodelers? Does ribosylation simply just sensitize nucleosomes to the remodeling reaction? So does it just weaken the DNA protein contacts? Or are there specific remodeler activation mechanisms? And so we dialed up a pretty well-established nucleosome remodeling assay, which is just based on restriction enzyme accessibility. It's called a restriction enzyme accessibility assay. And in this way, you can take your DNA template, you could put a PST1, restriction enzyme digestion site, into your DNA. And that PST1 site sits against your histone octamer. And so in this way, PST1 is unable to cut that DNA site unless your remodeler can shift the octamer down the DNA, exposing the digestion site and allowing the restriction enzyme to cut. Okay, and so if you perform an assay in the absence of a remodeler, you see no cutting of your DNA. And if you put a remodeler in, you could see time-dependent cutting of this DNA, which is representative of the remodeler translocating the nucleosome down that DNA strand. And so I'm just showing an example here of the CHD4 remodeler. And the CHD4 remodeler is insensitive to ADP ribosylation. It does not care. You can give it unmodified, you can give it our mono, or tri, or tetra, or doubly modified. It doesn't respond at all. I don't know if it will respond to far longer chains, but certainly in the context of our nucleosomes, this enzyme doesn't really seem to mind whether or not they're modified. This has kind of become the star of our show. So I just kind of wanted to give a brief background on this enzyme, ALC1. So ALC1 has been a pretty hot enzyme for several years now. And I think one of the more compelling stories about this protein came in 2017 from the Lerner lab in Germany, where he found that this protein, which has an ATPase domain, this is the domain that does the translocating, and it has a macro domain, which is known to bind to ADP ribose. So he found that if you put poly-ADP ribose, specifically tri-ADP ribose into this protein, it induced a large conformational shift that basically releases this macro domain from an auto-inhibited conformation and lets the ALC1 do its job. And then sort of the end of this paper is this model where here on the bottom left, you can see the macro domain kind of sitting over the ATPase domain, where auto-modified PARP, so this is PARP1 with that ADP ribose chain growing off of it, is able to bind to this macro domain and recruit the ATPase domain to chromatin to perform this reaction. And this has kind of been the generally studied activation mechanism because it's accessible, because you can just mix NAD and PARP1 together with nucleosomes and put ALC1 in, and you'll get remodeling. And so what we don't know is, how does this compare to nucleosome modification, right? So can nucleosome ADP ribosylation also induce ALC1 activity? And how do these things compare? And again, this is quite difficult to address in cells and even in a reconstituted setting without these technologies. So we found that indeed our ADP ribosylated nucleosomes are very efficient at activating ALC1. We see activation levels up to 350-fold with our doubly tetra ADP ribosylated nucleosomes. So really it's that tetra that kicks this off, just as would be predicted from the binding studies in the Lerner study. And that as the chains get longer, it doesn't really increase all that much. Site specificity doesn't really play in. It seems to just matter that you have this on that histone tail. And then we wanted to, again, compare back to this other activation mechanism, right? Specifically, how does nucleosome ADP ribosylation stimulate ALC1, right? How does this compare to how the auto-modified PARP1 stimulates ALC1? So we generated a bunch of auto-modified PARP constructs, things that had long and short serine ADP ribosylated chains, things that had long and short aspartate and glutamate ADP ribosylated chains. We performed a bunch of assays, and we saw kind of what had been reported previously, which is that PARP can stimulate these activities somewhere around 10 to 20-fold over unmodified. Again, whereas our nucleosomes were stimulating about 350-fold. And so we conclude that the nucleosome appears to be the preferred ALC1 activation mechanism. And another point that I like to point out, just to see what people think of it, is the nucleosome offers you a stable ADP ribosylation tethering site. And by that, I mean PARP1, when auto-ribosylated, falls off of DNA. That's kind of the main function for PARP auto-ribosylation, is to dissociate from DNA. And so you can imagine that PARP auto-ribosylation doesn't really give ALC1 a lot of time to come in and do its thing before it's going to diffuse away. Whereas nucleosome ribosylation isn't going anywhere unless this remodeler comes in and physically moves it. So there's certainly a lot of advantages in addition to this observed enhanced activity for this nucleosome ribosylation to be a real critical mechanism for activating ALC1. So I'm not saying PARP automodification cannot do it, but it appears that this nucleosome is a really good way to go. So we had a few more questions regarding specificity of this mechanism. And the first was, is ALC1 specificity maintained in heterogeneous nucleosome populations? And so by this, I mean, what happens if you mix an unmodified nucleosome and an ADP ribosylated nucleosome into one reaction, right? Will ALC1 still specifically pick one out, or once it's activated, does it kind of get sloppy and start modifying other things in the solution. So to do this, we assembled our different modified octamers on different DNA templates. Okay, so we basically gave these DNA templates a unique barcode that we could use to amplify in a qPCR reaction, right? So we could assemble our nucleosomes on unique templates, okay, mix them all together. So we could mix 5 or 6 nucleosomes together, be they mono, tri, unmodified, run our remodeling reaction, and then gather all of our DNA and run qPCR reactions for each nucleosome primer pair that we had. And so if we're destroying an amplicon, if we're cutting at our restriction site, we should not get amplification, right? Our CT values should go up. And indeed, that's what we see, that in these pooled nucleosome populations, we saw almost identical remodeling rates to what we were measuring in individual nucleosome assays. So ALC1 quite precisely targets these modified nucleosomes in this solution. And so the students said, that's not good enough. What happens on arrays? So they really wanted to do this assay where they assembled asymmetrically modified chromatin substrates. And it's a really great assay. I thought it would take a long time, but Jugal got this done in like 2 weeks, where he basically can make 2 different nucleosomes. Okay, in this case, one nucleosome A being unmodified, nucleosome B being modified. He can give these 2 sticky ends. And this has been done before. So this is not a new protocol. It's just new in terms of our ribosylation. But you can give these complementary sticky ends and ligate them together. And so in doing this, we now have dinucleosomes, right? Where we know that our remodelable nucleosome, or our nucleosome with the PST1 site, is either modified next to an unmodified, unmodified next to a modified, and so forth. Okay, so basically, the major question here is, can a modified nucleosome stimulate remodeling of an unmodified nucleosome directly next door? And so these are single time point assays. So I don't want to directly compare these levels the way that we did with rate constants. But the answer is that ALC1 activity is primarily confined to modified nucleosomes. So in the case where we have an unmodified nucleosome, right, that we're measuring remodeling on the unmodified, and it has our tetra ADP-ribosylated nucleosome directly next door on the same DNA template, we get very little enhancement over a totally unmodified dinucleosome construct. So again, this appears to be very specific. And then the last thing that we wanted to test was, does the loss of ALC1 impact this nucleosome ADPR dependent remodeling that we're seeing? And again, in cells, this is a very difficult question to address. So what we ended up doing was we created an ALC1 knockout cell line, and then we made nuclear extracts from either the knockout cell line or the wild type cell line. And we put our different nucleosomes one at a time into these extracts. And we just looked for, is there ribosylation dependent enhancement of remodeling activity? So what we see is that in wild type cells, okay, we'll always see a basal level of remodeling in these cells because there's lots of remodelers in these extracts like CHD4 that are going to remodel nucleosomes whether or not they have ribosylation. But when we add ribosylation to the substrates, that's these 2 here, right? These are the H2BS6 4X ADPR and the EE 4X ADPR constructs. We see this enhancement in remodeling activity. And when we take the ALC1 knockout lysates, we no longer see that enhancement towards these ribosylated substrates. So at least in the context of these extracts, ALC1 is required for this nucleosome ADP ribosylation dependent stimulation. And I think that together with those specificity experiments, what we're seeing is that this pairing of ALC1 and ADP ribosylated nucleosomes appears to be very precise. So there's lots of ribosylation happening at the damaged site, and there's lots of processes going on. But it looks like the ALC1 and the ADP ribosylated nucleosome are kind of made for each other. So I think that was very interesting. So again, this is our model. I'm kind of running out of time, so I'm just going to kind of breeze through this a bit, right? So we have that damage, priming, elongation, and remodeling. And I just wanted to touch on sort of why all this matters, because I brought this up in the beginning. But in the past several months, there have been quite a few papers showing that ALC1 is actually very effective, or at least knocking out ALC1 activity It's actually very effective as a combinatorial approach with PARP inhibitors. And perhaps more importantly, a lot of PARP-resistant cancers or PARP-resistant cancer models are responding very well to ALC1 knockout. And so now there is an emerging impetus, I've said here, for ALC1 inhibitors. And so we're hoping that by now understanding sort of some of the more basic principles of this ALC1 remodeling mechanism, that we might be able to feed into these inhibitor development efforts a bit. And then just, I'll just show you sort of two things that we're doing moving forward, and then I'll close off. So one is in a collaboration. So one of the authors on these papers, this Sebastian Diendl, he has a long history in studying ALC1 structure and function. I think a very clear move forward is to try and solve the structure of ALC1 bound to these ribosylated nucleosomes, right? That could certainly explain why we're observing this enhanced activity towards ribosylated nucleosomes over these other ribosylated molecules. And when we posted our manuscript to bioRxiv several months ago now, the Diendl lab reached out within a day or two, and they said that they had been trying to solve ALC1 nucleosome structures for a long time, and they've tried a bunch of different stuff. So mixing ALC1 and PARP and nucleosomes, trying to ribosylate nucleosomes with PARP and then solve the constructs that way. And that despite all these approaches, they're never able to get better than I think 5 or 6 angstrom resolution. And there's lots of heterogeneity. And clearly they think the reason is because, they're not controlling where the ribosylation is. They're not controlling things like chain length. And our nucleosomes could potentially really feed into their pipeline quite well. And so we just wanted to test before we'd send these out, can we make stable nucleosome ALC1 complexes? And the answer is yes. So what you're seeing here is a nucleosome on a native gel. If you mix in these ribosylated, nucleosomes and ALC1, you can see now 1-to-1 and 2-to-1 ALC1 complexes. You don't see these complexes if you use unmodified nucleosomes. They form just fine without cross-linking agent. When you add cross-linking agent, you kind of lock DNA down. So you get a little bit of a gel shift, but we're seeing very stable 1-to-1 and 2-to-1 complexes. And so at this point, rather than set up this pipeline, the Diendl lab seems to have this sort of all well and set up. We've sent these histones out to them, and they're working on this structure right now. So we're hopeful that this will answer some of the questions that we've had from our manuscript. And then the last part I wanted to touch on was, I want to skip that, just how can we study other PARP enzymes and substrates? And so there's lots and lots of PARP enzymes, and I think lots of open-ended questions here. And I don't have time to touch on a lot of it, but I think a good point here is a lot of these are known to modify aspartate and glutamate residues. And there's no methods for that. And I think pretty clearly, we have a chemoenzymatic approach for the serine. We thought, why not just get our hands on a bunch of enzymes that have been reported to target aspartate and glutamate residues. So, we went through everywhere we could find Addgene, the Lee Krause inventory, looked for all the enzymes that we could find that would do ASP-Glu. We purified them all. We tested them against a bunch of what we call a panel of substrates that have different possible modification sites, ASP-Glu, the serine, glutamines, etc., to look for ones that are very specific for ASP-Glu, but very promiscuous in terms of substrate specificity. So they won't hit anything except ASP-Glu, but they don't care about what's going on around them. And then taking some of those, anything that kind of matched this criteria, and optimizing that enzyme performance. And so no tricks here with the exception of consensus enzyme sequence design. And so if you don't know what this is, we'll take, and our example here is PARP14. Take PARP14 and get a bunch of homologs from a bunch of different species. We pulled from like 25 different species. You align those sequences, and then you take the most common residue at every site and you express and purify that. And this is a very well-established way to get enzymes that are very stable. And in general, because they're stable, can give you sort of more sustained activity over a greater period of time, higher yield, etc. So you probably see where this is going. We did find an enzyme that is able to modify any peptide that we give to it at aspartate and glutamate sites, and it is aspartate and glutamate specific. So now we're pushing that forward and thinking about things like antibody development, protein synthesis with those. And so with that, acknowledge my lab. So the ADP ribosylation contingent is Jagal here, who took over most of that story. Q here, who's working on sort of other activators and other ADP ribosylation sites, as we talked about on that last slide there. And then some other members of my lab are working on things that are not related to protein modification, just sort of other projects, but always helpful to one another. So with that, thank you for your attention. I'm happy to take questions.
Hey, Glenn, I had a question. So you identify serine S10 and S28 as ribosylated. But I find it also interesting that those are also phosphorylated, both during mitosis as well as S10 can be phosphorylated during interphase in some cells. So what's the relationship between like S10 phosphorylation and the poly-ADP ribosylation? If it's phosphorylated, does it block the ribosylation, or does it have to be reversed before the ribosylation occurs?
Yeah, that's a good question, right? I think, to me, sort of the thing that makes the most sense there would be that if you have DNA damage, particularly sort of a lot of DNA damage, ribosylation is a good way to make sure that phosphorylation doesn't happen, right? So now you're going to pause the cell cycle. Yeah. As far as I have tried to ribosylate phosphorylated peptides, it might work, but it's not the activity that we usually see. And I don't think the mark is very stable. So I think for my money, phosphorylation blocks ribosylation and vice versa. Okay. But yeah, I do think it'd be a good way to make sure the cell doesn't, progress through the cell cycle with a lot of damage around.
Yeah. So another question is, how long do the in vitro PARP1 reactions take in the assay? Also, what are the physiological concentrations of PARP and HPF, and what are your concentrations relative to that?
Yeah, okay, great. So in the reactions go quick. So PARP will torch 10 millimolar NAD very quickly. Usually like within 20 minutes, the reaction's over, whether you want it to be or not. And because it goes through so much NAD, it generates a lot of nicotinamide and then it's inhibited. So adding more NAD doesn't help your cause because you're just inhibited at that point. So our reactions generally 5 to 20 minutes when we do any of our, and this is just for the technology. So I'm not super worried about staying close to physiological. I just want to get the peptide ribosylated, but we are working at pretty short time scales. So the next question, part of this question is super relevant, which is what are the concentrations in cells? So PARP1 is present at roughly 10 to 20 fold higher concentration than HPF. So there's far more PARP than HPF. And there is a paper out now from Ben Black, University of Pennsylvania, on the bioRxiv, kind of looking at how does this work? So, how can such little HPF regulate the elongation activity by PARP, right? And I assume that's kind of where this question is headed. And it looks like HPF kind of rapidly associates and dissociates from PARP to basically tune chain length. But I think the whole story is not there yet. So it's very interesting that there's way more PARP than HPF. For us, the kinetics of that monoribosylation reaction and the elongation from that monoribosylation reaction, they both look very fast. It doesn't look like mono is way faster than elongation or vice versa. So I think there's a lot more work to be done as far as how this is regulated. And one sort of thought that we have is, I showed very early on that in a reconstitutive reaction, HPF gets ribosylated. And in cells, we see HPF gets ribosylated. And our thought is that potentially this HPF ribosylation helps to kind of break that complex. And so as the reaction progresses, you get a lot of monoribosylation happening at all these target proteins. And as HPF gets monoribosylated, it falls off. And now you have PARP to elongate from all those seed sites that HPF was there for. So there's a lot of possibilities for how this elongation reaction is regulated, but certainly the cellular concentrations are an important part of that.
So another question coming from Lee Krause. You're 2 for 2 on identifying ALC1 modulating serine residues. Is there specificity? So in other words, are there sites that don't modify ALC1 activity when ribosylated?
Yeah, so looking at, so in the nucleosome, right, you have the DNA entry exit site, which might be like my fingers, right? And then my thumbs are the back of the nucleosome. So the H3 tails come out, right, by the entry exit DNA. And then the H2B tails come out almost in the opposite direction going back. And they both activate just fine. And so my bet is that as long as you're on a histone tail, that you're going to activate fine. With that said, I was quite surprised that in the dinucleosome construct, that one tail, right, projecting out from nucleosome A was unable to stimulate on nucleosome B. Because if there's no great site specificity, why would it matter that it's on that nucleosome and not just an adjacent nucleosome, if that makes sense? I do think if you put this on a core, it might be a problem, because the ATPase domain kind of engages that core a bit more intimately. And I wonder if macrodomain binding and ATPase domain binding on the core of the histone would kind of be competing with one another. But my hope is that if the Dinda lab can get this structure information, we might kind of learn a lot more about this specificity.
All right, another question is, what is the functional difference between monoribosylation, di, and triribosylation?
Yeah, so you know, it's funny. There's a push now that mono is like the vast majority of what's happening in cells, and we've been studying a few things like chromatin compaction and other chromatin proteins that fall off of chromatin when they're ribosylated, and ALC1. And they're all poly. They all need poly to do their job. I haven't found a really good, in terms of DNA damage induced in PARP1, a really strong PARP mono ribosylation phenotype. I think monoribosylation is going to be useful. I'll use the HPF1 PARP interaction as an example. I said I think potentially ADP ribosylation on HPF1 breaks the interaction. I could imagine monoribosylation being a nice big mark that would disrupt an interface, right? So for something like that, where it's happening directly at an interface, I think it would work fine. But in terms of the difference of mono, di, and tri, that's really dictated by what the reader domain, what the interaction domain will recognize. So in ALC1, for example, it won't bind to mono or di, right? The Laderno lab has shown that, and we have found that as well with our ADP ribosylated molecules. So in that way, it's dictated by what the reader domains have evolved to bind.
So kind of extending upon that, like if you're looking, like you said, you've sort of defined this in the context of DNA damage. Like if you look at the different poly ribosylation chain lengths between DNA damage and then say look in the gene expression regulation kind of aspect of PARP, do you think you're going to have different dependencies on chain length between the two roles?
Yeah, I think that's probably going to be likely. And I say that because if you think about, if I think about some of the other proteins that have been implicated in ADP ribose dependent recruitment to chromatin, for example, a lot of them look like classic nucleic acid binding domains, right? Like RNA binding domains or DNA binding domains. And it looks like that interface, right? If you kind of do some structural predictions about the nucleic acid interaction motif, it looks like they're going to require 8 or 10 or longer, right? 8, 10, or maybe even up to like 12 residues, the same way you see with like a transcription factor, or, you know, a fuse or an H and RMP, like these RRMs and stuff. So my bet is they might require slightly longer chains. maybe they're less HPF1 dependent, right? So the chains get longer in those processes. But it's definitely an interesting direction to go.
Very cool. Great talk, Glenn. Everyone, thank Glenn for sharing this presentation with us, and thank you.
All right, thanks so much, Matt. Take care, everyone. Thank you.